Our last experiment is over proteins. You probably know that proteins are made up of amino acids, so let's start there. Here is an amino acid. It is both a carboxylic acid and an amine. Now I know that that carboxylic acid looks a little different because it's missing the hydrogen here. And that's just because at neutral pH, that hydrogen actually will float off. And the amine looks a little different too, but for the same reason, that there are hydrogens that can come and go, both one of these and then the one that's here, and they come and go depending on the pH. This is the real structure at neutral pH. And what's interesting is that at neutral pH, it's a Zwitter ion. A Zwitter ion is an ion with both positive and negative charges. Sometimes they're not drawn as Zwitter ions, they're drawn as I have down here, where you actually see the carboxyl group and the amino group, but technically that structure does not exist at any pH. It just makes it easy to see. Okay, so there are lots of different amino acids, and we say that there are 20 standard amino acids that our DNA codes for, and each has a different R group. That's what makes them different. So I'm talking about this R group here. The top part is the same on all of them. So if you look at these, you know, they all have this part in black the same. It's the red part that's different. The red part is the R group. Now you don't need to memorize any of the amino acids for me, but the the experiment involves a couple of the amino acids that you need to know something about what's on their R group. So glycine is the smallest one. You need to know that one. Its R group is just hydrogen. If we have a test that requires a certain group on the R group, like if it requires a phenyl ring or something, glycine is not going to have it because glycine is just a hydrogen. Okay, and then there are some more down here, some big ones. This one right here is called lysine. It has an amino group on its R group. That will react with some of our tests. These two both have benzene rings on them. We call that, the benzene ring, we call it as a branch, we call it a phenyl group. The rings. This amino acid is tyrosine. And this amino acid is phenylalanine. Okay, so those are the particular ones that will come up in the experiment. But do notice that different amino acids have different properties on their R group. In other words, like this one is totally nonpolar. This one's an acid actually. This one actually is basic. Lysine is basic. Some of them are big, some are small. These with the rings are really huge. Different properties on different amino acids, different flavors, chemical flavors. Okay, so a protein is a chain of those amino acids, and usually they're really long. Most proteins are in the thousands range. 2,000 amino acids linked in a chain would be a large protein. A lot of them, though, are in the 1 to 2 range. A small protein would be in the hundreds, something like that. A smaller chain of amino acids, which they do occur too, are often called a polypeptide instead of a protein. But those words are kind of used differently by different people, but generally peptide or polypeptide are for something small. Okay, this is a picture that shows you how the amino acids are linked together. The R groups are in red, and so you see the backbone is made from that black part that's the same on every amino acid, where we have here's our carboxyl group, and then this one's amine group had reacted with the next one's carboxyl group here. So that's what reacts when they form their peptide bond. One's amino group reacts with the next carboxyl group, and notice that once they react, it's not an acid anymore, and it's not an amine anymore either. Technically, this is an amide functional group. So anyway, so one end of the protein will have a carboxyl group, and one end will have what we call a free amino group. That it's not linked to something else. These groups in here are not called free amino groups. That comes up in one of the tests we'll see later. Okay, we know that proteins don't just stay in a long chain. Even the ones that end up kind of stretched out will be folded in a very specific way. So we say they're folded because we don't want to make it sound like they just get wadded up into a big glob, even though they kind of look that way sometimes. They're not wadded up randomly. They have a very specific shape. We call that specific shape their native state, their natural shape. This is the shape they need to be to work, to do their job, if they're an enzyme or a receptor or whatever they might be. To denature a protein is to do something to cause it to lose that native state. So if you denature a protein, it will no longer be active. And this is why we didn't want to heat up our saliva too much when we were doing our starch lab. If we had heated it up too much, it would have denatured our enzyme amylase, and then it would not have worked. Okay. There are different ways to denature. Changing pH is one of the ways, and we will use that way in lab. Let's look at more what happens when you denature a protein. When you denature a protein, it's not just that they unfold, they will stick to each other. Forming an insoluble solid. So here's a picture. Here's a protein that is native. This would be in solution, surrounded by water. Think of egg white would look like this, although the proteins don't look quite like these. I made these kind of small, but you can tell they're orderly, specific shape. Okay. If I heat it up or if I change the pH, they can start to unfold. And it's not just that they unfold, it's that when they start to unfold, they start touching each other. You see, parts of the protein that are supposed to be tucked in the middle start getting exposed, and they start touching the other proteins that are nearby. And when they start touching each other, when they start unfolding and touching, they will suddenly just collapse together, form one big solid. So this is, we say this is, they are coagulated. And they are denatured. So denatured means they're not folded correctly. Coagulated means they're stuck together. But those two things go together. This is a solid, like this. So we start here with something that looks, it may look like a slightly cloudy solution, because those are pretty big. But then when they start to unfold, they form a solid, like a precipitate. There are several diseases that involve misfolded proteins, because when they start forming proteins like this, these form plaques and they can kill cells and stuff. So Alzheimer's is one of the diseases, and mad cow disease, but there are many. Protein misfolding is even important in type 1 diabetes. When proteins start misfolding, they can cause real problems for cells.